Hello guys, welcome back to my channel. This video is only for your good preparation for the NEET exam. I will discuss regularly biology MCQs with you, and definitely it will help you to qualify the NEET exam. I appreciate each and every one of you. If you found value in this video, please consider liking, subscribing and sharing. Let's keep this community growing, and obviously also feel free to share your thoughts in the comment section. Now the question is, where will you look for the sporozoites of the malarial parasite? The answer of the options are salivary glands of freshly molted female Anopheles mosquito, saliva of infected female Anopheles mosquito, red blood corpuscles of humans suffering from malaria, and spleen of infected humans. Here the correct answer will be option 2, saliva of infected female Anopheles mosquito, because the mature infective stages of malarial parasite which are transferred from mosquito to human are sporozoites. The nucleus of oocyst divides firstly by meiosis and subsequently by mitosis, forming a large number of small haploid nuclei. At the same time, the cytoplasm develops large vacuoles. The tiny nuclei and cytoplasmic masses from elongated and spindle-shaped bodies called sporozoites. Each oocyst produces about 1000 sporozoites. When mature oocysts rupture, the sporozoites are liberated into the hemocoel of the mosquito. Being motile, the sporozoites move to different organs in the body cavity of the mosquito, but many of them penetrate the salivary glands of the mosquito. So sporozoites are found in the saliva of infected female Anopheles mosquito. When the female Anopheles mosquito bites a healthy person, the sporozoites are injected into his or her blood along with saliva. These sporozoites start the life cycle again in the human body. The next question, a certain patient is suspected to be suffering from acquired immunodeficiency syndrome. Which diagnostic technique will you recommend for its detection? The options are Vidal, ELISA, MRI and ultrasound. Here the correct answer will be option 2, ELISA, because ELISA or enzyme linked immunosorbent assay is a plate-based technique. It was the first screening test commonly employed for ELISA. It has high sensitivity. The common principle of ELISA is the use of enzyme conjugates that bind to specific HIV antibody and substrates or chromogens that produce color in a reaction catalyzed by the bound enzyme conjugate. So here the correct answer is the ELISA. Now the next question, select the correct statement with respect to disease and immunization. Options are statements are certain protozoans have been used to mass produce vaccine. Injection of snake antivenom against snake bite is an example of active immunization. If due to some reason B and T lymphocytes are damaged, the body will not produce antibodies against a pathogen. Option 4, injection of dead or inactivated pathogens causes passive immunity. Here the correct answer will be option 3. If due to some reason B and T lymphocytes are damaged, the body will not produce antibodies against a pathogen, because the immune response depends on B and T cells interacting with each other to provide an effective defense. T cells are the coordinators and they stimulate B cells to divide and secrete antibodies into the blood. These antibodies destroy on the antigenic pathogens, and the T cells then seek out and kill any of the body's own cells. Next question, read the following statements having two blanks A and B. A drug used for dash patients is obtained from a species of the organism dash. Here the options are there is two column blank A. There are 4 options for blank A and 4 options for blank B. The options for blank A are swine flu, AIDS, heart, organ transplant, and the options for blank B is Monascus, Pseudomonas, Penicillium and Trichoderma. Here the correct option will be organ transplant for blank A and Trichoderma for blank B, because we know Monoscarpus is an yeast from which statins are produced, which act as a clot buster and controls cholesterol level. So it is not related to swine flu. Pseudomonas is an infection causing bacteria. AIDS is caused by a virus. C. Penicillium is an antibiotic and is not related to heart patient. Okay. Trichoderma polysporum is a fungus and that is used to produce the drug cyclosporine, which is used to overcome rejection reactions after organ transplant. So here the correct answer is A, organ transplant, and B, trichoderma. Common cold is not cured by antibiotics because it is caused by a gram-negative bacteria, not an infectious disease caused by a virus, caused by a gram-positive bacteria. Here the correct answer will be option 3, caused by a virus. The common cold is a viral infection of the upper respiratory system which includes the nose, throat, sinuses, eustachian tubes, trachea, larynx and bronchial tubes. The most common groups include rhinoviruses and coronaviruses. Antibiotics are useless against a cold and can only enhance bacterial resistance if used without sincerity. Thank you. Stay tuned for our next video and I would truly grateful it if you enjoyed this one.